All right, well, continuing on with this discussion of methods, as I said, in the second part here, I want to talk about mathematical methods in this area called biometry. All right. So what happened here is that when Darwin proposed his ideas about natural selection and things, immediately, of course, the idea was put forward that it impact, humans were involved. And there had been an appreciation that humans were not a unique system to themselves. They weren't divinely different. They were in fact part of a natural system. Okay, and of course that was very controversial to many people, but it'd been an idea that'd been around for a long time. But Darwin kind of focused that in together. In fact, one of his later books was on human nature, right? Which because at first he didn't want to deal with this human part of it. But later on, he was kind of forced to, so he did it. But for us, what's important is that out of Darwin's work came really two big camps. Okay, one camp was the camp that looked at natural selection and said, we look at these things called sports, right? You're doing all these breeding experiments, and or you're raising things on a farm, and all of a sudden this thing with no horns shows up, or something with spots shows up. Everything else is solid colored, or everything else has horns. Those are called sports, and sometimes those are good, sometimes they aren't good. Sometimes that's led to, if I get rid of horns, they don't tear my fence down, so I'll start breeding more, try to breed more of these things that don't have horns, that are polled. This was called sports, and the idea was that these were kind of discontinuous evolution. Suddenly things just changed, suddenly things just appeared, and then those became very popular, or they had some advantage under Darwinian ideas, and they populated. Okay. And this fit in well with things eventually when the Mendelians came along, which, Darwin knew about Mendel's work, but he didn't really incorporate it. He was in the pangenesis. That was the best he could do for genetics. But at the same time, on the other side was a group that said, no, when we go out and look at things, they're not just everything different, right? We see small differences between things. We see things that can grow this height, and then something that can grow this height, and something grow this height. Not just one thing that's here and then one thing that's here of the same species. And so they looked at these small differences and said, what's selection really working on is these little small differences that occur. And some of the famous work, they were taking out people and lining them up and saying, look, human height, for example, is a curve, right? There's a bunch of people that are in the middle, and there's a few people that are very, really, really tall, and a few people that are really much shorter than the average in the middle, right? Excuse me. These individuals became known as biometricians. And biometricians were interested in this continuous system. And they fought. That's why this big arrow is here. They fought and fought and fought. In the literature, no, it's not that, it's this. And when Mendel was rediscovered in 1900, then they still fought for a while. And it wasn't really until 1918 that Sir Robert Fisher, put it all together. He came up with the concept of quantitative genetics, which we'll talk about, which led to the idea of population genetics. In other words, yeah, Mendel's right, okay? If you take this thing with a genotype and this thing with a genotype, and you mate them, you can predict what they're gonna produce, right? If you cross a heterozygote with a heterozygote, you can predict what they're gonna produce. Though, when you go out into a group of things, a population of individuals, well, sometimes you got het with het, sometimes you got het with homozygote, sometimes you got two homozygotes, you got the whole mix. Well, then you're going to see these distributions that occur here. And you put all that together in what was known as population genetics. And we'll come back to talk more about that on some other forms, okay? All right. Well, he wasn't the first biometrician, obviously, okay? There were lots of them around, and so I wanted to cover just a few of them that relate into human systems a little bit, okay? And into human genetics in particular, okay? So let me do that, okay? The first one I want to talk about was John Otterputnam, and he was an individual that sat down and had some really interesting ideas about human systems. One, he looked at about 80 years of data, of birth data from the UK, and said, oh, look at this. There's actually more males being born than females being born, okay? And so he actually derived the idea that there was a sex ratio, that it wasn't supposed to be well, potentially because of the mechanics that were thought of at the time and that we know today, you should get one male for one female at birth, right? But the reality is in humans that you don't, right? Some populations you get as many as 105 males to just 100 females. Now, if you go, of course, and look at in ecological time, you look over lifespan of those individuals, and you look at the average number of females and the average number of males that are alive when they're 80, okay, from that original cohort, then what you'll see is a complete transfer of that. Females will far outnumber the males on average, at least they have for the last few generations. Okay? But what was interesting is that he developed this really early idea of a p-value. He did what's known as a sign test, equivalent, that's what you call today, a non-parametric test, a sign test. He said, okay, I'm just going to count up the number of times it's a male, the number of times a female, and it should be 50-50, and it's not. He came up with a p-value. But now which we use all the time, okay? The test, right? You talk about the 95% confidence interval, or, oh yeah, I reject the hypothesis at this level. Well, that goes back basically to this individual. The other thing he did, among many things, one was that he was really interested in smallpox inoculation tests. And he actually did tests. He started out by inoculating, I think, his yard boy, but he went in and he actually looked to see, okay, if we inoculate people and we expose them and they get exposed to smallpox, what's going on? Now, smallpox had been a big deal, okay? I mean, smallpox had ravaged human populations, and it did ravage human populations up until the 1950s, okay? It was a debilitating, deadly disease for much of human history. There's good evidence from things like Egyptian mummies that there was smallpox available. It just, ravaged populations. Well, and it turns out that the Chinese, being very, very bright, about in the 1840s, or excuse me, 1540s, 1550, somewhere in there, there's writings from those periods that clearly indicate that they were doing inoculations. They were doing smallpox inoculations in China. Once China opened up, right, and ships started sailing around the world, they started bringing this information back to the Eastern world, and people started trying it out, right, and sort of working with it. Now, in 1722, by 1710 or so, they were in 1720s, they were inoculating people that were trying it. He actually did a smallpox test, an inoculation test, to see, okay, if I do this, what's the survival rate? Because in China, the survival rate was somewhere around, the death rate was somewhere around 2%. Okay, so if you got inoculated, because this is really crude, they don't have a dead virus or a dead pox here. What they were doing, it was taking a knife or something and mixing some ingredient and just poking you with it. And, you had about a 2% death rate. Now, it sounds terrible. If we were to make a vaccine today that killed 2% of the people, it wouldn't be very good. But remember, this is a early method, and it's certainly better than the 30 to 40% mortality that smallpox was causing. Okay, so why it might kill you, it certainly give you a better chance. It really wasn't until 1796 that an effective vaccine was produced, not in the sense that we think of today as a vaccine, but a way to grow it. It was actually produced from cowpox. There had noticed that his one of his maids or something had been milking a cow and got cowpox. It's essentially in the same family as smallpox. He also inoculated some yard boy or something as well, exposed them to nervous things, and they had found out that it worked. So it became a popular way to do it, right? And it actually had already been used in other places. This whole concept of inoculation, not necessarily the vaccine per se, but part of the success of the Revolutionary Army was the fact that Washington pushed to have these inoculations done. And they did some broad spectrum inoculations. Now, there was a lot of resistance to that, of course. But the reason they did it is that the Continental soldiers had lived many of them had been born in the United States, in North America. So those from the South or the North that were involved had been born here, had never actually been exposed to smallpox. Many of the soldiers that were coming in from England had been exposed as children and survived, and therefore they had lifetime immunity. And so smallpox was just ravaging North America once it came in. Boston and other places, I mean, there were just terrible cases of smallpox. And so Washington had actually, he was born and lived in Barbados. He had actually contracted it as a small child, and he knew how awful it was and how debilitating it would be to his army. So he actually started to have individuals checked. If they hadn't had smallpox, then they were, some of them were inoculated. One of the big places that it worked was at Valley Forge. We all know the story of Valley Forge and the horrible winter. Well, one of the reasons that the Continental Army actually survived there was, even though there was lots of smallpox and stuff, because when they had gone into the Quebec campaign and others, they had just been decimated by the smallpox, is that they did inoculations. And so even though it was a horrible winter and lost a lot of people, it would have been even worse had they not done this, okay? And then it was 1813, there was the Vaccine Act. The U.S. Senate, U.S. Congress passed the Vaccine Act. It was the first federal, we can think of in a form of a direct consumer protection and a pharmaceutical law. The reason it was passed is that there were a lot of these, they were doing these inoculations now around the country, and there was a supply problem. They had gotten some that were too virulent, and there had been some false stuff going on, and so there wasn't any control. And so they passed this law with certain stipulations about how the vaccine would be administered, and where it would be purchased from, make sure everybody had a clean supply of it. It was repealed in 1822 primarily so that the laws for this were pushed to the states, state instead of a federal mandate. But it's fairly successful while I was there. It's sort of limiting the amount of smallpox things. Now, of course, why am I bringing that up? Well, for other reasons, one smallpox, but people were anti-vaccine at the time, right? People just, nope, I'm not doing that. You're not sticking me with that. It's got, because so we think about the current situations and things that are going on with various diseases, and we have people that are called anti-vaxxers, right? They don't want the kids vaccinated. They don't want to be vaccinated. They're not going to do it. We're just going to rely on herd immunity and things. Well, yeah, herd immunity will work if you under the right conditions, but it takes a lot of deaths to get there. So this whole idea of anti-vaxxers is nothing new. It's been around. Probably it was in China, certainly was in the UK back 200, 300 years ago, and it was in the US and had been strong and running ever since this period of time. Okay. Well, that's a little side trip, but I wanted to kind of bring that up. All right, so there's a couple other people that related again to human systems. James Lidd was British scientist, and when the British Army, British Navy went out and sailed, they lost more of their sailors to scurvy than they did to both the Spanish Armada or any of the other French warships. In fact, there was one ship that went out with 1,900 men on it, and only 1,400 of them died over a couple-year period with scurvy. Lidd was studying this, was on a ship studying this, and realized, and there were multiple ideas about what might cure, deal with this. So he actually did multiple tests. He set up tests where he looked at people drinking vinegar and sea water and I forget all the other things, but a whole series, like 5 different treatments that he come up with, one of which was with lemons and citrus, right? So everybody got citrus, so much citrus a day. And it turned out that the people that were given the citrus recovered, okay? And that was a very small sample, but they recovered. All the other stuff, vinegar and salt water and other things, grog, they didn't recover, okay? And so he showed that, yes, we need to, so, carrying this, I think sulfuric acid was one of them, be a tough one to drink. But this was a concept of clinical trials, which are critically important in how we, once we identify something, whether it's a genetic, where we're trying to come up with a treatment for a genetic defect or for another, organic effect, and the idea that there can be multiple hypotheses that can be tested at one time. And then finally, of these individuals in history, there was Pierre Charles Louis. And what he did was he's thought of as he was French, obviously, thought of as the father of epidemiology. He was really coming up with what was known as numerical method. People at the time in the 1800s would just go and if they hear a treatment, they just try it, right? Poke people or do whatever. And they really didn't put any of the data together. Well, he would pull data together. That was his big thing. And look at kind of the average. Now, he didn't know about randomization and doing tests really, but he would pull the data together and say, oh, look, here's the average, or what kind of happens for the most of the time. Here's what the good side and the bad side is. One of the biggies that he looked at was pneumonia. In the 1800s, pneumonia was highly prevalent, right? There was no antibiotics for it. And so one of the common treatments was bloodletting. They go in and you have, and they all has bad humors, and they would cut you in bloodlet, and it had been carried on for a long time. And he looked at the results of individuals that had pneumonia and did have bloodletting, and people that didn't have bloodletting, and showed that bloodletting was actually making it worse. And so it was one of those things. He did the same thing with some typhoid fever and some other areas. So now it was one of those ones that led to this whole area that we have today. All right, now take us back directly into the genetics though, okay? We probably have to look at Carl Pearson. You may have all heard of the Pearson product moment correlation, or not, you will if you take statistics. He was a true biometrist. He was in biometry, a follower of Darwin. He developed a number of tests that we use today, the chi-squared test, that we taught in genetics lab that students just hate. Observe minus expected divided by expected, so you can figure out whether the ratio fits, and they always seem to be forgetting it. Little simple test for that, okay? Did it for genetics, all right, looking at a number of things. Come up with the concept of standard deviation. So while Luis had been looking at the average, okay, the question becomes, well, what's the spread away from that average? That's the standard deviation. And he was instrumental in the development of correlation and regression, although there were several people involved with these. Regression, the name regression, actually comes from looking at what's known as mid-parent rooting. So what you do is you take maybe something height. Okay, he was interested in height. He was one of the people that used one of those distributions of humans standing around with their height. And you would take the mid-parent. Okay, it's still a technique that's used around, but you would sit there and you'd say, okay, dad was 6'1", mom was 5'8". What's the average of those, mid range of those? And you would plot that against the actual height of the child. And what you saw is there was a relationship, okay? And that relationship, of course, we now know is a correlation. You're related, genetically on average. If your parents are both tall, on average, the children will both be tall. If the parents are of less height, the children will be of less height. And this was called correlation. Now, the regression part comes from the fact that regress back to the mean, right, to the average. That was the idea, and that was a big problem for Darwin, because think about it: if you developed a new mutation, okay, a new form, and it was a tall thing, who did it ever mate with? Well, shorter things. Well, then all the children should be somewhere down the middle. So you could never really get things to grow very much without it. It was regressing to the mean. Okay, we know now that that's not really how it works, but that's where the word regression actually came from. We regress back to an average form. Okay, well, these are all important people that were involved in sort of this mathematic methods and biometry. You'll hear more about them. We'll talk more about them. What I want to do in part 3 is now I want to move to actual methods. Okay, so I want to talk about what are some of the important methods. What ones might you see when we're in our reading, and why, what's good about them, what's not so good about them. All right, and so that'll take a whole lecture set on itself.